Hello students. The topic I would discuss here is post-mating isolation mechanisms and various theories related to origin of reproductive isolation for speciation. Gametic mortality. It is a post-mating pre-zygotic isolation mechanism. In these cases, gametic transfer occurs but fertilization of the male and the female gamete does not take place. The sperm becomes inviable or destroyed in the female reproductive tract. In plants, the pollens fail to germinate if they fall on the stigma of a different species. Since the gametes fail to unite, it is also known as gametic isolation. According to Kohn and Orr, the gametic isolation can be divided into two: non-competitive isolation and competitive isolation. Non-competitive isolation occurs due to poor transfer of sperms between different taxa, less mobility and inviability of the gamete in the reproductive tract of female of different species, failure to penetrate the egg to fuse with the nuclei, and reduced rate of oviposition. In case of competitive isolation, as the name suggests, competition occurs between the male gametes of different species in the female reproductive tract. If gametes of two different taxa are present in the reproductive tract of the female, the conspecific sperm or male gamete is preferred over the heterospecific sperm or gamete. In the absence of co-specific gamete, fusion of heterospecific gametes occur. Example in ground cricket. In many closely related species, under lab conditions, it is possible to overcome pre-mating barriers. However, the offspring produced are inviable or sterile. This is due to post-zygotic isolation. Post-zygotic isolation could be due to external environment factors, that is extrinsic, or due to internal factors, intrinsic. Extrinsic post-zygotic isolation is due to the interaction between the genotype and environment. And hybrid could be reared under controlled lab conditions, but when in nature fail to compete or are less fit. In general, an extrinsic reproduction isolation evolves before intrinsic reproductive isolation. Example of extrinsic reproductive isolation is the three-spine stickleback fish in Canadian lakes, wherein the hybrid cannot compete with the parents in their respective niche. Intrinsic reproductive isolation is due to genomic incompatibilities, which is manifested in hybrid inviability, hybrid sterility. It can be explained by Dobzhansky-Muller model and Haldane model. I will discuss it later in the session. Another way of post-zygotic isolation mechanism is zygotic mortality. Here, fusion between the male and the female gamete occurs, but the zygote does not occur due to genetic differences in them. Example, in sheep and goat, the zygote is non-viable. Next, hybrid inviability. In such cases, the zygote is formed, but the hybrid zygote fail to complete the developmental cycle to form the embryos due to abortion at various stages of development. This could be due to unfavorable interaction between chromosomes of two species, interaction of endosperm with the embryo, and incompatibilities between the cytoplasm and the nuclear gene. Example to this is hybrids between two different species of frog do not survive beyond the earlier gastrola stage. Another example to this is the salamander species where interbreeds may occur, but most of the hybrids do not complete the development cycle, and those who do look weak and frail. Another way of post-zygotic isolation barriers are hybrid sterility. In such cases, the hybrids are viable, but the resulting adults are sterile. It could be due to chromosomal incompatibilities during meiosis pairing. Example to this is mule, a hybrid of horse and a donkey. In certain cases, the hybrids formed are fertile, but their progeny are weak and sterile. This phenomena is known as hybrid breakdown. Example, interspecific hybrids of cotton and certain parotid wasp belonging to genus Nasonia. In this, it was studied that the F2 hybrids formed from two different species of Nasonia showed increased larval mortality rate relative to their parental species. Earlier, it was thought that it could be due to incompatibilities between the multiple nuclear loci and cytoplasmic factors of parental species. However, this could only account to 40% of mortality rate. Later studies found that the hybrid larvae carry incompatibility allele on chromosome 5, which was responsible for poor development of these hybrids and led to death of these hybrids before they reached adulthood. This slide summarizes both pre-mating and post-mating reproductive isolation mechanisms that are seen in organisms. Theories and different thoughts for origin of reproductive isolation. According to Darwin's theory, speciation occurred due to natural selection. It was later that the concept of reproductive isolation mechanism was brought by certain evolutionary biologists and zoologists like Dobzhansky and Mayer. Two mechanisms work for natural selection. First, ecological speciation, and second, mutation order speciation. In case of ecological speciation, the two populations get separated due to ecological or geographical barriers and get diverged by natural selection. In this process, selection acts in contrasting directions in individuals of the two populations depending upon the environment the two populations are, and leads to fixation of alleles. Example, in case of Darwin's finches. Note, in this case, the environmental pressure is different on the populations. In mutation order speciation, populations diverge as they accumulate different mutations under similar environmental pressure. Natural selection drives to allele fixation in both these methods. However, in the first case, selection favors divergence only under ecological speciation, while in mutation order process, divergence had occurred in the population by chance. In a diverging population, therefore, isolation mechanisms are required which would prevent the exchange of gene pool between the diverging population. Genetically conditioned intrinsic mechanisms help in this separation of two diverging species. Therefore, the origin of reproductive isolation mechanisms are important mainly in case of populations which are under the same environmental pressure and share common distribution. Reproductive isolation mechanisms increase the efficiency of mating by preventing wasteful courtships. However, if the two populations are ecologically separated, an additional isolation mechanism is not required for divergence of the two populations into two different species. How is reproductive isolation established between populations? There are different schools of thoughts. According to Alfred Wallace's theory, natural selection selects isolation mechanism. The healthy progenies, that is, the ones with superior genotypes, are selected against the weak hybrids in a population. In such a scenario, pre-mating reproductive isolation mechanisms are induced or enhanced by natural selection, while post-mating isolation appears as a byproduct of divergence. As per the theories put forward by Edward Poultron, Muller, and Mayer, reproductive isolation is a byproduct of genetic divergence, and speciation is incidental. According to Haldane theory, hybrids which are formed, the one with this heterogametic sex would be inviable, weak, and sterile when compared to the homogametic sex. Always, hybrids with heterogametic sex have lower fitness and hence evolves faster than homogametic hybrid sterility. Postzygotic isolation follows the Haldane rule and heterogametic gender evolves faster. Muller's view on reproductive isolation mechanisms. According to Muller, reproductive isolation is an accidental byproduct of divergence in an allopatric population. The population first splits into two due to a geographical barrier. The two subpopulations are acted on by different evolutionary forces, example mutation, selection, and genetic drift. Based on the advantage of certain mutations and genes, these alleles get fixed in the population with time. This leads to reshuffling and remodeling of the genetic constitution in both the subpopulations. If, in case, individuals of these populations come together after a very long period of time, hybrids are not formed because of their disharmonious genotype. This is an illustration of Muller's view of speciation and reproductive isolation. In this, reproductive isolation starts as an incidental byproduct of genetic divergence and is completed and promoted by natural selection. Multiple gene hypothesis or multiple allele substitutions has also been proposed to explain the origin of reproductive isolation. This is known as gradual speciation. In case of an allopatric population, the reproductive isolation is a byproduct of speciation mechanism, while in a sympatric population, reproductive isolation is reinforced into the system. The hybrids that are formed between closely related species are often inviable or insterile. This is due to the chromosomal incompatibilities that happen in the animal in the meiosis stage. It could be due to change in ploidy also. The mechanism of post-zygotic isolation can be explained by Dobzhansky-Muller model. According to this model, post-zygotic isolation is a result of interaction between genes or alleles at multiple loci rather than genotype at single loci. This leads to hybrid incompatibility. Hybrid inviability is caused due to the phenomenon of underdominance at a single loci. That is, homozygosity at a loci has an advantage over heterozygosity. Individuals with example capital A and small a are viable while hybrids with capital A small a are non-viable. The simplest way to explain Dobzhansky-Muller model is by using two loci, A and B. The ancestral genotype is considered to be capital A, capital A, capital B, capital B. That is homozygous dominant alleles at both the loci. Due to some reason, the population split into two, population 1 and population 2. In these two populations, the selection pressure due to environments are different. With time, a mutation occurred at allele A loci for population 1. So, capital A becomes small a due to this particular mutation. This mutation turned out to be advantageous for the population 1, and natural selection helped in fixing this allele into population 1. So now The genotype of the individuals in population 1 becomes small a, capital B, capital B. Similarly, in due course, mutation occurred in loci B for population 1 and it gets fixed in the population by natural selection. Individuals in population 2 have the genotype capital A, small b. Now, when individuals in population 1 and population 2 form a hybrid between them, both the loci turn out to be heterozygous. The hybrid has a genotype of capital A, capital B, small b. These hybrids would be weak or sterile or non-viable. This is because the interaction between the two loci, small a and small b, are incompatible. The small a allele is advantageous in a capital B body, but not with small b allele. It is a epistatic interaction. Allele at A locus are compatible, that is capital A and small a, and allele at B locus are also compatible, capital B, small b. Thus, postzygotic isolation is due to multi-locus genetic interaction. The DM model or Dobzhansky-Muller model can be explained by an example. Example, hybrids formed between two closely related species of fish, platyfish and swordfish. The platyfish has spots on their dorsal fin, while the swordtail lacks this spot. In a backcross hybrid, that is F1 hybrid mated with sword-tail fish, some progeny had large spots. The one that is seen in the extreme left, which in turn out to be a malignant one. This reduced the lifespan of these fishes. Please study the phenotype and the genotype of each individual. Explanation to this is the platyfish with spots have a X-linked gene XMRK2, a receptor kinase that is responsible for producing spots, and also an autosomal repressor that checks the expression of spot producing gene. The sword-tail fish lacks both XMRK2 gene, that is spot producing gene, as well as its repressor. In backcrosses hybrids, some of the hybrids receive the spot producing gene from the platyfish but not the repressor gene. These individuals are the ones that developed malignant tumors because the expression of spot producing gene is not regulated. This is an illustration of a hypothetical example in case of salamanders as to how sibling species of salamander diverge to form a new species by means of prezygotic and postzygotic isolation mechanisms at various stages and levels. To summarize, evidences suggest that reproductive isolation accumulates more rapidly in a allopatric population than in a sympatric population. Genetic and ecological isolation may occur at the same time or before or after reproductive isolation mechanisms. Evidence or factual evidences point towards the rarity of speciation without a previous geographical isolation. Not all reproductive isolation mechanisms are required for any particular speciation event, and its sequence of development also varies from species to species. The figure on the right shows two populations which evolve under different environmental pressures and evolutionary forces. When brought together, if they fail to form a hybrid, they are known as prezygotic isolation. If the hybrid is formed but are non-viable or not sterile, it is known as postzygotic isolation. Thank you.